Now it's time to talk about protein synthesis. How does your DNA control the production of your proteins? Central to this discussion is the knowledge of RNA. So you have to know something about RNA. RNA stands for ribonucleic acid. It's a little different than deoxyribonucleic acid in that it has an extra oxygen in each ribose molecule that deoxyribose does not have. Otherwise, they're the same except for uracil. I'll explain more of that in just a second. RNA is a single-stranded molecule, so it's kind of like a half double helix. It's only one rope of nucleotides with nitrogen bases sticking out, and that's what you see here in this diagram. DNA is actually, remember, double-stranded, two ropes, and the nitrogen bases actually are complementary to each other. The adenine and thymine are complementary, the guanine and cytosine are complementary, and that allows the two sides to stick together because their shapes match and they hydrogen bond to each other. So RNA is only single-stranded, and it's got a different sugar in it. It doesn't have deoxyribose; it has ribose instead. Lastly, RNA has cytosine, just like DNA cytosine. It has guanine, just like the DNA's guanine. It has adenine, like the adenine of DNA, but it does not have thymine. Here's thymine. It has a substitute called uracil. Now, these two things are almost identical. The difference is this is called a methyl group. Thymine's got a methyl group. Uracil is missing the methyl group. But uracil otherwise does exactly the same thing that thymine does. It bonds to adenine whenever necessary. So, why did I bring the RNA up? Well, it turns out that DNA dictates what proteins you make, but it does not, and your ribosomes actually make the proteins, but your DNA does not touch your ribosomes. Instead, it sends out RNA copies of itself, which is what the ribosomes then use to produce proteins. Step one of protein synthesis is called transcription. That's the production of RNA. So transcription is step one in the overall process of protein production. It's the process by where a molecule of DNA is copied into a complementary strand of RNA. Okay? DNA remains in the nucleus. It does not go out to the ribosomes. The enzyme that does this is called RNA polymerase, and it builds RNA by attaching to DNA, and then it reads the nitrogen bases on the DNA, and it builds a complementary copy. RNA polymerase knows how to do this because it attaches to a special start area just upstream of a gene. That's called a promoter. We'll talk more about that later. And that serves as a start signal on the DNA for the RNA polymerase to attach. So your chromosomes are not just random adenine, guanine, cytosine, thymine ropes. They're specific coded regions. And each section of your chromosomes that are responsible for making a protein is called a gene, Gene. Just upstream, meaning towards the 3' side on the DNA, is the promoter, which is where RNA polymerase attaches. And then like a train, it just slides down the track. And when it gets to the start of the gene, it transcribes RNA from the DNA by reading it. So if this was the green right here is an RNA transcript of DNA. Notice the G matches the C, the A matches the T, the C matches the G, the U matches the A. RNA has no thymine. It has uracil as a substitute. This is RNA polymerase, and it's sliding down to the right. And it's reading the DNA, and it's producing an RNA copy of the DNA. That's how transcription works. This type of RNA is usually called messenger RNA. It's built by RNA polymerase from the DNA, from the start of a gene to there's actually an end message at the end of the gene to tell RNA polymerase where to stop. That's called transcription, the production of messenger RNA from gene sequences in DNA is transcription. Now, how is the genetic code written in your chromosomes? You're going to need to know how to read this codon wheel here. The DNA information is in a code, and it's sort of an alphabet-based language where A represents adenine, G represents guanine, C represents cytosine, and T represents thymine. There are, obviously 20 different amino acids to represent in the genetic code, but it turns out there are 64 different words possible in DNA. So each DNA word, as it turns out, is 3 nucleotides. So AAA is a genetic word, GGG is a genetic word, CGA is a genetic word. Each codon specifies a specific amino acid. Some amino acids are represented by up to 6 codons, and a few are only represented by 1 codon, like tryptophan and methionine only have 1 codon to represent them. There are 3 stop codons that do not represent amino acids. They stop protein production. They signal the end of a polypeptide. The scientist that figured out the entire genetic code was Marshall Nierenberg, and in 1968 he got the Nobel Prize for working out the translation table that I'm going to show you how to use now. So when DNA produces RNA, the RNA is actually a strip of nitrogen bases, but to a ribosome, the RNA nitrogen bases are read 3 at a time. If an RNA codon reads GAG, what amino acid would the ribosome be putting together there? Well, GAG stands for, according to the codon wheel, reading inside out, GAG stands for GLU. GLU stands for glutamic acid. That would be the amino acid that the ribosome would be putting in position when it gets the RNA codon GAG to work with. If instead it got the RNA codon CGA, CGA stands for arginine, or ARG. Over here it tells you that it's arginine. So as a result, the amino acid that would be strung together when a codon CGA is read by a ribosome is arginine. Now, there's one last piece of information before we start into the specifics of protein production. There are special worker molecules that bring amino acids to the ribosomes, and they are called transfer RNAs. Transfer RNAs have anticodons, this is the anticodon, and they carry amino acids on the opposite side. So this is more like the shape of the tRNA, but we tend to draw them as a cloverleaf for simplicity's sake. So the amino acid is carried on the opposite side of the anticodon. Why do I bring this up? Well, as it turns out, anticodons and tRNA molecules are critically important for bringing amino acids to the ribosome so that the ribosome gets the proper amino acid when it needs it. There are actually 3 kinds of RNA. Well, there's more than that, but there's 3 basic kinds of RNA found in cells. There's transfer RNA, and that's what you see pictured here. There's messenger RNA, we already talked about that. And then there's ribosomal RNA, which we haven't talked about, but that makes up just about 50% of the mass of a ribosome. Now, one more time, what transfer RNAs do is they have cloverleaf shapes. They have anticodons that determines the type of amino acid they will carry, and they carry amino acids to the ribosomes because their anticodons must base pair with the codons on messenger RNA to bring an amino acid to a ribosome. So translation is the actual process of protein production. If you recall from the cell chapter, ribosomes are made of 2 subunits, a large and a small, and each one is made up of ribosomal RNA and proteins. Now, when the ribosome assembles, one side of the ribosome is called the A site, where new amino acids keep getting brought in, and the other side is called the P site, where the polypeptide that is being made, is held. Now, translation is initiated when the small subunit binds to the start codon region on the messenger RNA. I'll show you this in an animation in just a minute. So, first I'm going to show you an overall animation, then get to the specifics. So let's go over the overall animation here. Let's wait for it to start. In fact, let me just go to the one where I can control it. Now, here's a nucleus. There's the nuclear membrane. Here's a chromosome, little piece of DNA. These are ribosomes. Okay. These are amino acids, and these are transfer RNAs. The objective is to show how proteins get made. There's no protein in this cell. There are amino acids, but there are no proteins. The first step of protein production is always transcription. So there you see, this is transcription up here. You'll notice that only one side of the DNA is being transcribed. The other side is silent. It does not transcribe. And that's because it doesn't have a promoter on its side. Now, RNA polymerase moves down the DNA and it produces a copy of the DNA called messenger RNA. That's what this is. This looks like a comb. That's a little messenger RNA molecule. That will slip out of the nucleus through one of those big pores, and then a ribosome will attach to it. Now, this shows it attaching to the ribosome, but it's generally the other way around. Then, transfer RNA molecules that have the proper anticodon plug will lock on to the messenger RNA, bringing their amino acids. The ribosome's job is to put these little amino acids together, and that's what it does. It keeps locking them together. You'll notice the little balls are getting locked together. Okay? Now, at the end of this, when all the little balls are locked together, notice that the tRNAs came out. Now, let's back up a second. See the free amino acid sticking out here? The tRNAs, after they dropped off an amino acid, they went out and picked another one up, and they brought it back to the ribosome, and they drop them off. In the end, when you get to the end of the messenger RNA, now you have a polypeptide. This is a protein right here. Could be, that could represent keratin, which is in your hair, or collagen, which is in your cartilage and bone, or it could represent growth hormone, or hemoglobin, which is the red pigment in your blood, or any of the other thousands of proteins that are present in the human body. Now, here it is in color, same animation. Here's the messenger RNA at the top. It slips out of the nucleus into the cytoplasm, drops down onto a ribosome. Notice the little amino acids have different colors because they're different amino acids. The tRNAs bring them in, drop them off on the ribosome. The ribosome strings the amino acids together by condensation. We talked about this. This is condensation here, not hydrolysis, condensation of amino acids together, and we get a finished protein. Now, here it is more up close and specific. So let's look at the one I can control. This is zoomed in now. Now you're looking at a piece of DNA inside the nucleus of a cell. The hand is RNA polymerase. It pops open the little chromosome here, and it reads the nitrogen bases on the DNA, which start with TAC, and then GCT, GTA. It's going to read all the way down, and it's going to put RNA nucleotides together. Now notice the RNA nucleotides have little purple sugars because their sugars are ribose and not deoxyribose. So the RNA polymerase moves down the line, and it puts little nitrogen bases in sequence, little nucleotides in sequence. It just keeps moving down the line, and what it's doing is it's making a string of RNA. Okay, that string eventually will reach its end when the RNA polymerase reaches the stop signal. This is the stop signal right here, conveniently at the end of the chromosome. At that point, this is messenger RNA. Notice that the DNA is still double-stranded. The A's and the T's balance out, and the C's and the G's balance out. Notice that the top strand is facing left because its 5' is on the left side. The bottom strand is facing right. Its 5' strand is on the right side. Notice that if you remember, the RNA polymerase was on the bottom strand. It reads from 3' to 5', and it did not read the top strand at all because there was no TAC on the top strand. Now, the RNA polymerase then, the RNA polymerase and the DNA stays in the nucleus, but the messenger RNA goes out one of those big pores and slides out in the cytoplasm. Then the small subunit, this is the small subunit of the ribosome, attaches. Then the first transfer RNA, now it's plugged, the anticodon has to match A, U, G. U matches A matches U, C matches G. That transfer RNA carries methionine all the time. When that locks in, the large subunit attaches. At that point now we have a ribosome complex on the messenger RNA with the first transfer RNA locked in. Then the next transfer RNA locks in, its GCU matches CGA. The ribosome then puts a bond between these two amino acids. It sticks them together. Then the first transfer RNA leaves. Okay, this is the A site right here. This is the P site. Watch how this works now. The ribosome will shift down one. This is the P site. The left side of the ribosome is the P site. The right side is the A site, because the right side is where the new amino acids keep getting brought in. The next one, the tRNA is GUA matching CAU, and it carries histidine. The ribosome bonds the two amino acids together right there. The previous transfer RNA is no longer needed, so it slips away to go pick out another amino acid, like it's got to pick up another arginine. Then the ribosome shifts the A site down so that the next tRNA with the anticodon UGC will match and lock onto ACG, and that'll be bringing threonine. The ribosome bonds those amino acids together. The process keeps repeating. The movement of the ribosome is called translocation. So the ribosome translocates. The A site is now open for the next tRNA. The next tRNA comes in. It's got an anticodon ACG, which matches UGC. It's carrying cysteine. It locks in. The ribosome bonds the two together. And now we've got 5 amino acids strung together. The next tRNA locks in, UUG anticodon matching the messenger RNA AAC codon. The ribosome locks those amino acids together. Then this is the last amino acid comes in. ACC matches UGG, anticodon to codon. The ribosome bonds the asparagine to the tryptophan. At that point now, the ribosome shifts down and it reaches UGA. Now, UGA is one of the 3 stop codons. That tells the ribosome, you're done. At that point, then, the ribosome cuts, the large subunit cuts away, the small subunit cuts away, and then the polypeptide will roll up. It may have to go to a Golgi apparatus to be modified, but this is the polypeptide manufactured by transcription and translation of the original DNA gene. It's just a string of amino acids, and that's what the human body is. It's proteins, which are strings of amino acids doing amazing things in human body activity. So there's a summary up here. The first tRNA carrying methionine will always bond to the first mRNA codon. The first mRNA codon will always be AUG because the first DNA codon is always TAC that's in the beginning of a DNA gene. The large subunit will then attach to the messenger RNA, and that's after the first tRNA and the small subunit has attached. The ribosome bonds amino acids together. The first tRNA that came leaves, the ribosome translocates, the A site waits for more new amino acids to be brought in, etc., etc. Ultimately, the polypeptide will either work right off the ribosome or it'll be sent through the ER to a Golgi body to be modified and then activated. This is another artist rendition. The ribosome's the big green thing. The mRNA is the greenish strip here. And the tRNAs are the yellow things carrying the amino acid balls. The A site is over here where new tRNAs are coming in, and the P site's on the left where old tRNAs are leaving. Now that concludes our discussion of protein synthesis.